Welcome to this 8th lecture in this course on eukaryotic gene expression. In the last class we talked about the importance of chromatin in the regulation of gene expression, and we discussed about how covalent modification of histones can affect gene regulation, and we specifically discussed one particular post-translational modification of histones, that is acetylation and deacetylation of histones, how they influence gene regulation. And I told you very clearly that there are enzymes called as histone acetyl transferases which add a acetyl group on the histones, and this acetylation of histones results in a positive regulation of gene expression or activation of gene expression. And there are also enzymes called histone deacetylases or HDACs which remove the acetyl group from the histones, and this results in the repression of gene expression. So today what we will do is we will go and then discuss one more post-translational modification of histones, namely methylation, and then see how methylation of histones affects gene expression. And we will also talk about other histone post-translational modifications, especially phosphorylation and so on and so forth. So just to recapitulate what we have discussed so far in this course, we began with discussing about eukaryotic RNA polymerases. Then we discussed about some of the core promoter elements like the Tata box, the initiator, and so on and so forth. Then we talked about general transcription factors which come and assemble in the core promoter elements, and we in fact discussed how variations in general transcription factors itself can bring about differential gene regulation, and variations in the core promoter elements can also bring differential gene regulation. Then we moved a little bit further upstream and then discussed about enhancer sequences and upstream activation sequences. Then we discussed how transcriptional activators, which are sequence specific DNA binding proteins, go and bind through their DNA binding domains specifically to specific sequences in the upstream regions, and then through their transactivation domains can influence the rate of transcription initiation. And just recently we discussed about DNA is not naked inside the cells. DNA is actually organized in the form of chromatin. You have nucleosomes, and therefore the first thing that has to happen if transcription initiation has to take place, the DNA has to be unwound, the histones have to be displaced, and this is where covalent modifications of histones have become a very important player in regulation of gene expression. And in the last class we discussed about how acetylation and deacetylation of histones regulates gene regulation. So acetylation is one major mechanism by which gene expression and gene transcription can be activated in eukaryotic cells, and today how we'll discuss how addition of a methyl group to histones bring about changes in the gene regulation, and we'll also talk a little bit about phosphorylation of histones. So the enzymes which will add acetyl group are called histone acetyl transferases. The enzymes which will add methyl group, which we are going to discuss today, they are called as histone methyltransferases, and enzymes which phosphorylate histones, they are called as histone kinases. So as now just mentioned, let us discuss about methylation of histones and their role in gene regulation, and the lysine and arginine residues of the histones can be methylated by proteins known as histone methyltransferases. So the two amino acids, lysine, which are both are basic amino acids, these are the targets for methylation, and the enzymes which will add methyl group to these histones, they are known as histone methyltransferases. So this is just a cartoon, just tells you how does the lysine residue of a histone look like, and you can see you have a positive charge here. That's what they call basic amino acids, and if you add one methyl group to this lysine, then it becomes a monomethyl lysine. So the hydrogen here is replaced by methyl group. Now if you add two methyl groups, then it becomes dimethyl lysine, and if you have three methyl groups, then it becomes trimethyl lysine. So you have histones. If you have only methylate one methyl group, it becomes a monomethyl histone, dimethyl histone, and trimethyl histones. Accordingly, depending upon the number of methyl groups that have been added to the histones, there is a nomenclature. I will just give an example here. For example, if you say H3K4Me3, it actually means in the histone H3, the lysine 4 residue is trimethylated. So lysine residue 3 of the histone H3 is trimethylated, and you denote this as H3K4Me3. Similarly, if you say H3K9, it actually means the lysine 9 residue of histone H3 has been methylated. So there are enzymes which will add only one methyl group, two methyl groups, or three methyl groups, and accordingly you have monomethyl, dimethyl, or trimethyl lysines in the histones. Now, the important distinction between the histone acetylation and histone methylation is that histone acetylation most often results in transcriptional activation. Okay, of course in biology there are always exceptions. When I say histone acetylation results in transcriptional activation, there may be one or two instances where histone acetylation may also lead to repression of transcription, but in general acetylation of histones results in activation of transcription. That's what happens in common. But unlike histone acetylation, the histone methylation can either result in activation or can result in repression of transcription. And here, for example, I have given an example where methylation of this particular arginine or lysine in the histone H3 can actually result in activation of transcription. On the other hand, if you methylate these lysines, it can actually result in repression of transcription. So, depending upon what kind of residues, which residue is getting methylated, it can either result in activation of transcription or it can result in repression of transcription. So, now let us study the enzymes which actually add this methyl group to histones. So, these enzymes which add a methyl group to histones, they are known as histone methyltransferases. Now, just as I told you that there are either a lysine can be methylated or an arginine can be methylated, and accordingly you have two classes of histone methyltransferases. They are called histone lysine methyltransferases HKMTs or histone arginine methyltransferases or HRMTs, or in general protein arginine methyltransferases. Now, let us now look at first histone lysine methyltransferases and see what kind of methyltransferases are there which methylate the lysine residue of histones. Now, the first histone lysine methyltransferase which was discovered in Drosophila is called SUV39H1 somewhere in the year 2000. So, this was the first histone methyltransferase to have been discovered, and this methyltransferase was soon found to methylate specifically the lysine 9 of histone H3. So, this particular methyltransferase, the SUV39H1, specifically methylates the lysine 9 residue of histone H3, and once people realized that the methylation of histones can actually take place and there are actually enzymes which can methylate histones, people started looking for such methyltransfer activities, and several other methyltransfers were later discovered, and we will discuss some of them in the next few minutes. Now, so all these studies have actually shown there are at least 5 lysine residues in the histone H3 which can be methylated by different enzymes, and these include lysine 4, lysine 9, lysine 27, lysine 36 and lysine 79. So, at least 5 lysine residues in the H3 are targets for 8 different methyltransferases in the eukaryotic cells, and in the case of H4, at least 1 lysine residue, that means the lysine 20, can be methylated by a specific methyltransferase. So, you can see here I actually designated the 3 methyltransferases which can methylate the specific histone residues in histone H3. The lysine residues are shown in pink color here, and you can see a methyltransferase called TRX in the case of Drosophila, and its homolog called MLL methylates the lysine 4 residue of histone H3. Similarly, the methyltransferase called HCV39H1 in the case of human, or SU329 in the case of Drosophila, they can methylate the lysine residue in the of H3, and the lysine 27 of histone H3 can be methylated by EZH2 in the case of humans, or its Drosophila homolog called EZ. So, there are specific histone methyltransferases which can specifically methylate specific residues in the amino terminal regions of the histone H3. Now, just as I told you in the last class that the acetylated histones are recognized by proteins through what are called as bromodomains, right? The same way the catalytic domains involved in methylation of histones in the case of histone methyltransferases are called is known as a set domain, and a set actually stands for the catalytic domain present in these 3 proteins S, E, T. So, when you say set domain is nothing but a catalytic domain in which is present in the histone methyltransferases that is involved in methylation of lysine residues of histones. This is called as a set domain. Now, histone methylation is traditionally linked to repression, but it can also lead to transcription activation. That is what I told you in the previous slide. Just as histone acetylation is most commonly results in transcription activation, in the case of histone methylation, although traditionally methylation results in transcription repression, there are instances where histone methylation, lysine methylation can actually lead to transcriptional activation. For example, I have given one example here. Lysine 4 methylation by a histone methyltransferase called SET1P can actually result in activation of transcription, whereas lysine 9 methylation by SUV39H1 actually can result in repression of transcription. So, histone methylation can either lead to activation or repression of transcription. In fact, when they have looked at this SET1P, which is a lysine methyltransferase, very often the SET1P was found to be associated with a HAT complex, that is the histone acetyltransferase complex. And now you can see how acetylation by SET1P can result in transcription activation. So, what could be happening is when this particular methylation takes place, this actually becomes a signal for histone acetylation, because set 1p is part of the histone acetylation. So set 1p probably will be bringing the hat complex to the vicinity of the methylated residue, and then the hat will now methylate other lysine residue, other residues in the histone H3 that can result in the activation of transcription. Very interestingly, the lysine 9 methylation in the case of histone H3 often results in transcription repression, and in fact it is found to be one of the major mechanisms by which euchromatin is converted to heterochromatin. So in most of the cases, in the heterochromatin, the lysine 9 of histone H3 was found to be methylated. Okay, and very interestingly, methylation of H3 and lysine 4 residue of histone H3 impairs methylation at lysine 9. That means when lysine 4 gets methylated by set 1p, the chances of lysine 9 getting methylated by SWIM1 is reduced. So methylation at this residue actually affects the methylation at the adjacent residue. So therefore, when most probably, that's why you always find lysine 4 methylation mostly in the euchromatic regions, because in the lysine 4 when it gets methylated, there are very rare chances that the lysine 9 will get methylated, and therefore very rarely you find heterochromatin when lysine 4 is methylated, because this actually reduces the efficiency of methylation of lysine 9 residue. So heterochromatin usually does not contain lysine 4, and therefore lysine 4 methylation is usually found in euchromatin regions, whereas lysine 9 is usually found in the heterochromatin regions, and methylation of lysine 4 actually negatively regulates methylation of lysine 9 residue. Now people have now looked at these methylated histones in a number of systems, eukaryotic systems like Drosophila, mouse and so on and so forth, and they have actually drawn certain conclusions, and some of these conclusions are: dimethyl H3H4 was shown to be associated with euchromatic regions. So when the presence of the 2 methyl groups in the lysine 4 is often found to be present in the euchromatic regions, and similarly trimethylation of the lysine 4 residue of H3 often is a signal that these genes are going to be actively transcribed. So it is not only the specific residue of lysine, but also depending upon the number of methyl group that is added can be a signature for a specific physiological response. So dimethyl lysines often is found in euchromatic regions, and the presence of one more lysine, namely trimethyl lysines, usually signifies that these promoters in which the particular histone has been contains a trimethyl lysine at lysine 4, these genes are likely to be actively transcribed. And the initiation and maintenance of heterochromatin is carried out by histone H3 lysine 9 methylation. So remember, whenever we talk about histone H3 methylation, there are only 2 residues which are very important. One is lysine 4, another is lysine 9, and lysine 9 methylation often results in activation of transcription. Lysine 9 residue results in the repression and heterochromatinization of the chromatin. So mammalian histone methyltransferases, which can methylate lysine 4 of histone H3, a number of such enzymes have been discovered, and some of them are mentioned here, and the names are right here. So a number of histone methyltransferases are capable of adding a methyl group in the lysine 4 residue of histone H3. And very interestingly, methylation of the lysine 27 has also been shown to be involved in transcription repression. So in addition to lysine 4 and lysine 9, lysine 27 is also plays a very important role in the regulation of gene expression and chromatin structure, and lysine 27 methylation can also result in transcription repression. So remember, when somebody asks you about how heterochromatinization, how histone methylation affects heterochromatin formation, you have to remember that methylation of lysine 9 or lysine 27 in histone H3 can result in heterochromatin formation and transcription repression. So methylation of these 2 residues can actually go into formation of heterochromatin. And H3 lysine, the lysine 27 methylation in the case of H3 histone is actually catalyzed by a multimolecular complex and is often referred to as the polycomb repressive complexes. Polycomb is the name of the protein, and this protein is actually involved in transcription repression and heterochromatin formation. We will discuss some more details about this in the next few more classes later, but just remember here that lysine 27 as well as lysine 9 methylation signals heterochromatinization, and methylation of lysine 27 is actually carried out by a huge multiprotein complex, and one of the members of this multiprotein complex is a protein called polycomb. Now, just as the acetylated histones are recognized by proteins which contain a specific domain called as bromodomains, similarly methylated histones are recognized by proteins which contain specific domains called as chromodomains. So remember, whenever we talk about histone modifications, chromatin and gene regulation, acetylated histones are recognized by proteins through their bromodomains, whereas methylated histones are recognized proteins through their chromodomains. So methylated lysines are recognized proteins containing specific domains known as chromodomains, and these are found in several chromatin associated proteins. So you can see here the way the whole system works is the moment an acetyl group is added, certain proteins containing bromodomains come and interact with these acetylated residues, and that is a signal for recruitment of other protein complexes resulting in transcription activation. Similarly, methylation of lysine residues in the case of histone recruits proteins which contain what are called chromodomains, and these proteins then bring other players into the field, and then depending upon which kind of lysine is methylated, it can either result in transcription activation or transcription repression. So the chromodomain specifically binds to the histone tails bearing methyl lysine. So it's very specific for methyl lysine residues, and the chromodomains have the highest affinity for trimethyl lysine and the lowest for monomethyl lysine. So if when you have 3 methyl groups on the lysine, the chances of it attracting a chromodomain containing protein are much higher than when only 1 or 2 lysine residues are methylated. A number of such proteins which contain chromodomains have been identified, and some of the most well studied are a protein called heterochromatin protein 1, now abbreviated as HP1, and in the case of Drosophila, a protein called polycomb protein plays a very important role in gene silencing and heterochromatin formation. In the later stages of this course, when we talk about gene silencing and other heterochromatination and so on and so forth, we will discuss the exact mechanism why these proteins actually bring about gene silencing and heterochromatinization, especially things like X chromosome inactivation and so on and so forth. But at this time point, just remember proteins, chromodomain containing proteins, the two very well studied examples are heterochromatin protein 1 or HP1 and polycomb protein or PC. Both HP1 and PC specifically recognize the lysine 9 and lysine 27 of histone H3. So the recognition of these proteins of these particular methylation groups result in transcription repression and heterochromatin formation. So I just now gain a cartoon here just to give an idea how exactly heterochromatin formation is actually initialized, because this results usually heterochromatin involves more than one type of histone post-translational modification. Usually, let us say for example, there is a euchromatin region, and I told you very clearly in the euchromatin region usually the lysine residues are in an acetylated form. So let us say for example you have a euchromatin in which the histone H3, the lysine 9 and lysine 14 are acetylated by a specific HAT, histone acetyltransferase. Now if a heterochromatinization has to be initiated in this particular locus, what happens? The acetyl groups have to be removed, and therefore a histone deacetylase now removes the acetyl groups from this particular histone lysine 9 and lysine 14 residues, and this deacetylated lysine residues now serve as the signal for recognition by a histone methyltransferase called SUV39H1. So the deacetylated lysine 1 and deacetylated lysine 14 now is recognized by a histone methyltransferase, and this histone methyltransferase now specifically methylates lysine 9. And methylation of this lysine 9 now attracts a chromodomain containing protein called HP1. Now HP1 comes and binds to this lysine 1, and then through subsequent methylations and multimerization of HP, heterochromatinization now spreads. So this is how entire chromosomes can be inactivated. So you can see the initiating event in heterochromatinization is deacetylation, followed by methylation, and then recognition of these methylated histones by chromodomain containing proteins, resulting in a chain of events leading to heterochromatinization of the particular region. So you can see how two post-translational modifications act in a coordinated manner to bring about a specific physiological response. In this case, deacetylation followed by methylation of specific lysine residue can result in heterochromatinization. So I just listed here the heterochromatin formation in one particular case results first involves deacetylation by histone deacetylases, then methylation by a histone methyltransferase, then recognition of this methylated lysines by a chromodomain containing protein like HP1, then initiation of heterochromatization, and then further propagation of this heterochromatinization by self-association of HP1. So I just mentioned another cartoon, the same example, where to actually show how specific histone post-translational modifications can either result in transcription activation or transcriptional gene silencing or heterochromatin formation, depending upon what kind of post-translational modification is involved. In these particular examples, for example, lysine 9, lysine 14 acetylation can actually result in the recruitment of a specific TAF, subunit of TAF, the TBP associated protein subunit 250, TAF2 250, which through its bromodomain recognizes the acetyl groups of these two lysines, and this now recruits other components of the general transcription factors, and RNA polymerase and transcription activation takes place. On the other hand, if this lysine 9 is actually methylated, instead of recruiting a bromodomain containing protein, now this now becomes a signal for a chromodomain containing methyltransferase, and then this chromodomain containing HP1 protein now recognize this methyl containing lysine, and then it brings about heterochromatin formation and gene silencing. So in one instance, the same lysine residue when acetylated now becomes a target for recognition by a bromodomain containing transcription activation, and the same lysine residue when it is methylated becomes a target for a chromodomain containing protein, resulting in gene silencing and heterochromatin formation. So you can see how just the modification of histone can actually either result in transcriptional activation or transcriptional repression, depending upon what kind of proteins are recruited to this particular modified lysine residue of the histones. So this is where the chromatin structure and histone post-translational modifications can have a very significant influence on the regulation of gene expression in chromatin templates. So far we discussed about histone lysine methyltransferases and how they affect gene regulation. Now let us talk briefly about histone arginine methyltransferases, which specifically methylate arginine residues of histones. At least two well-known histone arginine methyltransferases have been characterized. One of them is called as the coactivator associated arginine methyltransferase, another is called as the protein arginine methyltransferase 1 or PRMT1. These have been very well studied. Now, all these arginine methyltransferases or protein arginine methyltransferases contain a particular domain called as PRMT domain, which refers to protein arginine methyltransferase domain. Okay. The catalytic module that methylates specific arginines is known as PRMT, and this PRMT domain actually transfers the methyl group from S-adenosylmethionine to the guanidine group of arginines to produce monomethylarginine or dimethylarginine. Now methylation, for example, it has been shown that methylation of the arginine 3 residue of histone H4 can facilitate H4 acetylation and enhance transcription activation by nuclear hormone receptors. Now remember, here in this initial series of lectures, I am actually introducing you to specific concepts. Now later in the classes, we will revisit each one of these post-translational modifications. We are going to take specific examples. For example, when I say nuclear hormone receptors, we are going to spend at least 1 or 2 hours in the later stages of this course to actually discuss about how transcription activation is brought about nuclear hormone receptors, how when a nuclear hormone receptor binds to a specific target sequence of DNA, what kind of histone modifications take place, what kind of proteins are recruited, and what is the exact mechanism by which transcription activation is affected by nuclear hormone receptors, and so on and so forth. So we will discuss more and more detail the specific mechanisms by which the transcriptional activators bring out histone covalent modifications leading to either activation or repression of transcription. But what we are right now studying is actually kind of a foundation for you to understand some more advanced concepts of eukaryotic gene regulation. Now I am going to give one more interesting example just to give you an idea how important is this histone methyltransferases can be in certain specific physiological responses. Now let me give one example where people have actually shown that histone methylation plays a very important role during development, especially early embryonic development. For example, there is a mammalian histone methyltransferase known as G9A, which specifically methylates the lysine 9 of histone H3. Okay. And it has been shown that G9A actually represses the transcription of many genes involved in early embryonic development. Okay. So methylation of lysine 9 of histone H3 by G9A in the promoters of can actually bring about repression of a number of genes in the case of which are actually play a very important role in early embryonic development. So if you want to now really ask the question, how important is this particular methyltransferase? What is the physiological significance of methylation of lysine 9 of all these genes? Because many of these experiments are actually done in vitro, so just because a protein is getting methylated or phosphorylated in vitro by a specific enzyme doesn't really mean that it has a really physiological response. So to really understand whether this methylation by this particular enzyme really has any meaning or any physiological relevance, people actually knocked out this particular enzyme, this particular histone methyltransferase, and when you knock out this particular methyltransferase, you can such kind of a mice show severe growth retardation and early lethality. So they die at a very early stages of embryonic development. So there are embryonic lethals. Similarly, if you knock out this G9A in embryonic stem cells, embryonic stem cells are very, very important. They are the pluripotent stem cells which ultimately give rise to all kinds of tissue during the development. And if you knock out this G9A methyltransfer embryonic stem cells, those stem cells you actually can demonstrate there is a very, very reduced lysine 9 methylation of the histone H3, indicating that G9A is the dominant methyltransferase that specifically methylates the lysine 9 of histone H3 in vivo. So, it is a major enzyme, one of the major histone methyltransferase that specifically methylates this particular lysine residue, and if you knock out this particular enzyme, there is a drastic reduction in the lysine and methylation of histone H3, clearly indicating that this enzyme has a very important physiological role, and if you knock out this, the mice have a very, very severe phenotype. So, in the absence of G9A, H3-9 methylation was drastically reduced in the euchromatic regions, indicating that euchromatic histone K9 methylation regulated by G9A is actually essential for early embryogenesis. So, you can see how we both by using a combination of in vitro experiments where you actually demonstrate the enzyme specifically methylates a particular residue, and by actually doing experiments in whole animal models by knocking out, you can clearly demonstrate that this particular histone modification by a specific enzyme has a very high physiological significance because you get a specific phenotype. And this entire information is actually taken from this particular journal. So, the question now is, just like we have histone deacetylases which remove the acetyl groups added by histone acetyl transferase or HATs, similarly if you are adding a methyl group histones, there should also be enzymes which remove the methyl group. So, just as we have deacetylases are there demethylases. Now, for a long time people did not identify histone demethylases. In fact, many people thought histone methylation is a very, very stable post-translational modifications and probably it is a kind of a permanent phenomenon. But soon histone demethylases were indeed discovered. In fact, the first histone demethylase which was discovered is an enzyme called LSD1, which stands for lysine specific demethylase. So, demethylation by LSD1 is limited to either mono or dimethylated lysine 4 of histone H3, H3K4, and LSD1 cannot demethylate trimethylated H3K4. So, we still do not know once a trimethyl lysine is formed, how is it demethylated, we do not know still. This particular enzyme can only demethylate mono or dimethylated lysine 4 of histone H3. And there are examples to actually show this demethylation by this enzyme actually has a important role in gene regulation. For example, in the case of androgen receptor, which is a member of the steroid hormone receptor family and is a transcriptional activator, androgen receptor goes and binds to specific sequences called androgen receptor response elements, and binding of this androgen receptor to the response elements results in activation of specific genes. And androgen receptor was shown to alter the specificity of LSD1 from H3K4 to H3K9, and by this particular mechanism it converts a demethylase from a repressor to an activator of transcription. So, in general, demethylation by LSD1 of thing can actually result in repression, but in the presence of androgen receptor, the LSD1 instead of demethylating H3K4 now demethylates H3K9, and this actually results in activation of transcription rather than repression of transcription. So, you can see the same enzyme can change its substrate specificity in the presence of another protein. Normally it only demethylates H3K4, but in presence of androgen receptor it can actually demethylate H9, and these two can elicit totally different kind of physiological responses. Now, if you start now looking at some of the literature and journals, you will find there are a number of such examples where, although we describe here only about certain basic phenomena, there are variations of many of these phenomena. The same enzyme, depending upon the physiological situation, depending upon its interacting proteins, can change its specificity and can bring about different physiological responses. So, in addition to LSD1, once LSD1 was identified as a histone demethylase, many other demethylases were also discovered. For example, a new class of histone demethylase enzymes containing what is known as a Jumanji domain has now been identified. So, you have to now remember some of these nomenclatures which are very, very important. Bromo domain, chromo domain, set domain, Jumanji domains, all these domains are very, very important. They have very, very important functions. Some are for recognizing specific histone modifications, and some of them are very important catalytic domains of key enzymes involved in histone modifications. So, a new class of histone demethylase enzymes containing a new catalytic domain called Jumanji domain have also been identified. There are at least 15 such proteins which contain this particular catalytic domain, and they specifically demethylate either a lysine or arginine residue of histone H3. So, one of such enzymes called JHDM, which actually stands for JMJC containing Jumanji domain containing histone demethylase 1, specifically demethylates H3 at lysine 36. And another such histone demethylase called MJD2A is a lysine trimethyl specific demethylase that converts trimethylated H3K9 or H3K36 into dimethyl, but not to mono or unmethylated products. So, a trimethyl lysine is converted to dimethyl lysine, but whether this dimethyl lysine will further get converted to mono or methyl or unmethylated is not known. So, there are demethylases, just as we have deacetylases which remove the acetyl group added by histone transferases. There are demethylases which specifically remove the methyl groups added by histone methyltransferases. So, I have just given the whatever I have told you so far, I have shown in the form of a cartoon. For example, in the case of histone, the residues that are normally methyl are H4, lysine 4, K9 and K27. These are residues which are normally undergoes methylation, and the enzyme which demethylates lysine 4 is called LSDA1. The enzyme which demethylates lysine 9 is JMJD2B, and the enzyme which demethylates lysine 27 is JMJD3. Okay. So, we have so far discussed two important post-translational modifications that happen for histones. One is the histone acetylation, another is the histone deacetylation, histone methylation, and we also discussed there are enzymes which actually remove acetyl groups, and these are called as HDACs or histone deacetylases, and there are enzymes which remove the methyl groups, and these are called as histone deacetylases. Now, we will now talk about one more histone covalent modification, namely histone phosphorylation. So, specific serine or threonine residues of histones are phosphorylated by specific histone kinases. Okay. So, just as people discovered histone acetyltransferases and histone methylases, people have also identified histone kinases which specifically add a phosphate group to specific serine residues in various histones, and I just listed here some of the serine and threonine residues which are found to be phosphorylated in histones. For example, in the case of histone H2A, the serine 1 and threonine 1119 have been shown to be phosphorylated. Similarly, in the case of histone H2B, serine 14 and serine 32 can be phosphorylated in vivo. Similarly, in the case of H3, serine 3, serine 10, threonine 11, serine 28, tyrosine 41, threonine 45. Remember, there are 3 amino acids in proteins which can be phosphorylated: serine, threonine and tyrosine. Okay. So, the enzymes which phosphorylate serine is called serine kinases, serine protein kinases, threonine protein kinases, and there are enzymes which phosphorylate tyrosine residues called tyrosine protein kinases. So, similarly, in the case of H4, S1 and very rarely histidine also can be phosphorylated, and in one case, for example, the histidine 18 of H4 was also found to exist as a phosphorylated state, and so is histidine 75. So, histidines, serines and threonines in selective histones have been shown to be phosphorylated, but what are the actual enzymes which phosphorylate histones have not been identified or not been characterized very well. So, the histone acetylation and histone methylation and the enzymes doing these functions have been very well characterized, but the kinases which are actually phosphorylating histones have not been very well studied. Now, just to tell you that histone phosphorylation also has very important physiological significance, I am just giving you a couple of examples here. For example, in the case of Drosophila embryo during development, threonine 119 phosphorylation of histone H2A was shown to occur during mitosis, but not in the S phase of the mitotic cycle, and this threonine 1919 phosphorylation was shown to be brought about by an enzyme called nucleosomal histone kinase 1. So, histone phosphorylation has been documented in specific stages of cell cycle in Drosophila embryonic development. Similarly, in the case of H3 phosphorylation at T45, this was shown to occur after activation of DNA damaging signaling pathways, and this is mediated by protein kinase C. So, histone phosphorylation is not only involved in gene regulation, it is also involved in a number of other physiological responses like cell cycle regulation, DNA damage signaling and so on and so forth. Similarly, phosphorylation of tyrosine 41 of histone H3, which is actually carried out by an enzyme called Janus activated kinase or JAK2, and this has implications in hematopoiesis and leukemia. This is just an example to tell you that phosphorylation of this particular histones can actually result in the modulation of expression of genes involved in hematopoiesis, because if you now tinker with this particular phosphorylation, it can actually result in leukemia. In fact, people have actually shown that if there is a decreased phosphorylated tyrosine residue of histone H3, this actually results in the expression of an oncogene called LMO2. Now, LMO2 is a very important oncogene, and if you overexpress LMO2, it results in leukemia. In fact, in some of the gene therapy trials, when they tried to actually cure a disease called severe combined immunodeficiency syndrome by what is called as retroviral gene transfer, people have actually found out that this gene actually goes and integrates near this LMO2 gene and resulting in activation of this LMO2 gene, and as a result some of these patients which actually underwent gene therapy for severe combined immunodeficiency syndrome, they actually develop leukemia. So this LMO2 is a very important oncogene, and if you activate this oncogene or if you overexpress this oncogene, it can result in leukemia, and histone phosphorylation has been shown to play a very important role in the modulation of expression of this LMO2, and this LMO2 gene has a very important role in normal hematopoiesis. Similarly, histone H4 is phosphorylated on serine but also on histidine 18 and 75, but unlike the acetylation, deacetylation, methylation and demethylation, somehow the histone phosphorylation has not been very well studied, and the actual kinases which are actually involved in this histone phosphorylation are not that well characterized compared to the other post-translational modifications. Now, if you actually go into the literature, you realize that all these post-translational modifications, what I told you, either it is acetylation, deacetylation, phosphorylation, methylation or deacetylation, they have all been very well studied. In fact, today the literature is flooded with examples of all kinds of histone modifications that are taking place and what are the important physiological responses. In fact, there are very nice websites. In fact, there are companies which actually sell antibodies which specifically recognize a specific post-translational modification of a specific histone. For example, there are antibodies which will specifically recognize proteins in which the lysine 4 is acetylated, or lysine 27 is acetylated, or a specific lysine residue is methylated, or a specific lysine residue is phosphorylated. So using these antibodies, people have actually identified what kind of genes are associated with these particular post-translational modifications. For example, there is a company called Abcam which actually sells many of these antibodies against these peptides which contain specific post-translational modification of histones, and they give very nice charts, what is called as a histone modification charts. They are actually telling, and these are constantly updated, and these charts actually tell you what are the important post-translational modifications of histones which have been studied so far, and what are the enzymes which are actually involved in this, and what kind of transcription factors have been shown to be associated with this particular histone post-translational modification. For example, in this particular chart, it actually shows you what are all the acetylations of histones which have been reported so far in the case of yeast. For example, H2A, H2B, H3 and H4, you can see here lysine 14, which are all the transcription factors or coactivators which are known to be recognized involved in the lysine 14 acetylation. Similarly, GCN5, which is being shown to be involved in the recognition of lysine 18, and so on and so forth. So I am not going to the details. Basically, to tell you that there are now information available in literature where you can actually look up and see which are all the residues in various histones which are acetylated in species like yeast, mice or humans. This chart just tells you what kind of residues are acetylated in yeast. Similarly, residues which are phosphorylated in the case of yeast histones are shown in this chart. Residues which are methylated of different histones are shown in this chart, and residues which are ubiquitinated. Ubiquitination is a signal for degradation of proteins. Okay, and ubiquitination of histones also play a very important role in regulation of gene expression, and residues involved in which are specifically ubiquitinated are also shown in this particular chart. Similarly, there are charts which clearly tell you which are all the acylated residues in the case of H2A, H2B, H3 and H4 in the case of mammalian histones. Similarly, phosphorylated histone residues in mammalian histones, ubiquitinated residues in mammalian histones, and which are the residues which are methylated in mammalian histones, and so on and so forth. So what I have been discussing so far is, at least I have discussed about 3 major mechanisms by which the regulation of gene expression can take place. We began telling you that chromatin structure can be modified by specific post-translational modifications of histones, and post-translational modification of these histones can have a very important role in regulation of gene expression. And I mentioned to you that one of the most important or one of the first histone post-translational modifications which have been identified is the histone acetylation, and in fact one of the first histone acetyl transfer which have been discovered is a protein called GCN5P. And histone acetylation is brought about by a group of enzymes called histone acetyl transferases, and acetylation of histones can actually generally results in the activation of transcription. Similarly, once acetylated residues histones are acylated, there are a group of enzymes called histone deacetylases which actually remove this histone acetyl groups from this histone, and these are known as HDACs or histone deacetylases, and these HDACs actually play a very important role in deacetylation or repression of transcription. And then we discussed one more post-translational modification of histone, namely histone methylation, and I told you there are enzymes called histone methyltransferases which specifically recognize lysine residues or arginine residues of histones and then add 1, 2 or 3 methyl groups. Accordingly, you have a monomethylysine, dimethylysine or trimethylysine lysine containing histones. And methylation, unlike acetylation, can either result in activation of transcription or repression of transcription. And there are also enzymes called demethylases which remove these methyl groups, and again depending upon the situation, methylation or demethylation can either result in activation or repression of transcription. And histone methylation plays a very important role in heterochromatin formation or gene silencing. And we also recognized that there are proteins, there are specific sets of proteins which specifically recognize either acylated histones or methylated histones, and the proteins which recognize acylated histones have very important domains called as bromodomains, whereas proteins which recognize the methylated lysines or methylated residues of histones contain what are called as chromodomains. Okay. So, the histone post-translational modifications serve as signals for recognition of specific proteins through which contain specific domains, and we also discuss specific catalytic domains like set domain and so on and so forth, which play a very important role in their enzyme activity. Then I discussed about histone phosphorylation and how histone phosphorylation can also play a very important role. Now, what all these things actually told you is that histone covalent modifications has now brought in a new paradigm into the regulation of gene expression, and all of you so far must have studied what is known as a genetic code. Now, what is genetic code? Genetic code is nothing but information which is encoded in the DNA. Okay, the 4 alphabets A, T, G, C, and the way the codons are organized, the way this A, T, G, C sequence is read in triplets is what is known as a genetic code, and this genetic code is actually forms the basis for what we are today. That is the major mechanism by which information is transmitted from one generation to another generation. Now, once people have realized that in addition to the genetic code, in addition to the DNA sequence information, there also seems to be certain signatures or certain information which is encoded in histones. And I can all that we have discussed in the last 2 classes is basically specific covalent modifications of histones can actually alter gene expression, and therefore there seems to be some kind of a code which is also embedded in the histones, and this is what is known to be known as histone code. So, just as we have a genetic code which is determined by the linear sequence, DNA sequence, now we have people now begin to advocate a new theory that just as you have a genetic code, there may probably be a histone code which actually specifies the specific post-translational modifications that can ultimately have specific signature patterns of regulation of gene expression. So, just as you have a genetic code, there may be a histone code. So, what is this histone code? This histone code basically tells you that modification of histone tails represents a novel code that is different from that encoded in the DNA sequence. So, this information that is encoded in the DNA sequence is called as the genetic code, but covalent modification of the histones may also be a new code, and depending upon what kind of covalent modification takes place, you can actually specify or say what kind of genes may be regulated, what kind of gene expression changes may take place, or what kind of physiological responses may be elicited. So, there is and from 1 or 2 examples I have given clearly, it is very clear that there is an extensive talk between covalent modifications. There have been examples where I told you methylation of one particular residue affects the methylation of an adjacent residue, and there have been examples where, and I also told you, for example, in the case of heterochromatin formation, first the acetyl group has to be removed, and the deacylated lysines now serve as a signal for the recruitment of a methyltransferase, and then this methyltransferase now adds a methyl group to this deacylated lysine, which is then recognized by chromodomain containing protein, and this ultimately results in the gene silencing and methylation and subsequent methylation and spreading of heterochromatin. So many a times a combination of histone covalent modifications together can lead to a specific physiological response or a specific signal for activation or repression of gene expression. So one modification of histone can either enhance or suppress another modification. This is what I just discussed now. Many times the deacetylation, acetylation, methylation, demethylation, phosphorylation, they are all linked together. One modification of histone can actually lead to another modification, or one histone modification serves as a signal for another histone modification to take place, and it is the combined effect of all these post-translational modifications that may serve as a signal for a specific event. It can ultimately result recruitment of either activators or activation of transcription, or it can result in the repression of transcription. And I also told you very clearly that the modified histones serve as binding sites for specific protein domains. So once histones undergo post-translational modifications, these modified histones now serve as signals to attract specific proteins which interact with the specific modified histones through specific interacting domains. And I gave you two examples where proteins which contain what are called as bromodomains, they specifically identified acetylated histones, and this usually results in the recruitment of histone methyltransferases which results in the activation of transcription. Similarly, there are proteins which specifically recognize the methylated histones, and these proteins which recognize methylated histones contain what are called as the chromodomains. Now, although we discussed these things, and although we discussed very briefly about enzymes which phosphorylate histones, the exact mechanism by which these phosphorylated histones are recognized proteins is not very clear still. So what kind of kinases are actually involved in histone phosphorylation, and what kind of proteins actually recognize these phosphorylated histones is not very clear. So there are three important things that I wanted to remember when we talk about histone covalent modifications. One is the genetic code is embedded in the DNA sequence of an organism. It is the sequence of 4 letters ATGC, namely the DNA sequence, which constitutes the genetic code. Whereas in the last two classes we have discussed what is called as a histone code, and this histone code depends on the post-translational modification of histones, and this histone code is written by proteins which can be histone acetyltransferases, histone deacetylases, histone methyltransferases, histone demethylases, or histone kinases. Okay. So just as the histogenetic code is written by the linear sequence of DNA, the histone code is written by enzymes which post-translationally modify, which bring about post-translational modification of histones. And the histone code, once you have this modified histones, once a histone code is written, this code is now read by specific proteins, and these proteins include the proteins such as those which contain chromodomain proteins, or proteins which contain the bromodomain proteins. So this kind of histone code hypothesis, now this is still not very conclusively proven. Genetic code is very conclusively proven, but the histone code is still at a very early stage, and people think that just as you have a genetic code, and depending upon what kind of a linear sequence you can actually predict what kind of a protein it codes, what kind of a physiological function it may have, and so on and so forth. Similarly, people are now advocating there is something called a histone code. So people are now saying, depending upon what kind of histone is getting acetylated, or what kind of residue in histones is getting phosphorylated or it getting methylated, you can actually, that can have a specific physiological function, and this particular post-translational modification of histones can actually serve as a code for a specific physiological response. So this histone code hypothesis has now become a key to understand how eukaryotic transcription apparatus interacts with chromatin. So you can see now we have come a full circle. We started looking at gene regulation as if DNA is naked, and we started looking at what is happening at the core promoter sequences, what is happening at the upstream promoter sequences. Then we discussed about what kind of protein factors go and bind near the core promoter elements, and what kind of protein factors go and bind to the upstream activation sequences. And then I very briefly discussed about the link between activators and the general basal transcription machinery, and I told you there could be proteins called as co-activators which may actually act as a bridge between activators and the RNA polymerase machinery. Then I deliberately did not discuss this co-activator concept in detail because I thought I should bring in the chromatin concept first, because the major mechanism by which all the gene expression changes are taking place, the chromatin seems to be playing a very important role. And now in the last two classes I have described how covalent modification of histones has become a very important player in the regulation of gene expression, and we have now reached a stage where people are now proposing that there is actually something called histone code. Okay. So, just as you have a genetic code, probably there is a histone code, and depending on what kind of post-translational modification is taking place for a particular histone, a specific physiological response may be emanating. And I gave you at least half a dozen examples where if you block a specific post-translational modification of histone by knocking out a specific histone methyltransferase or knocking out a specific histone acetyltransferase, you can actually bring about specific changes. And in fact, people this see the topic I have chosen for this particular course is eukaryotic gene expression basics and benefits. Okay. Now this histone modification has now become the basis for the founding of a number of pharmaceutical companies. A number of pharmaceutical companies are now working to see whether you can develop actually inhibitors which can specifically block a specific post-translational modification of histones. Okay. For example, I will come back and revisit some of the topics which I mentioned earlier. There are companies now, for example, which are working to develop small molecules which specifically block the activity of histone deacetylases. And in fact, people have actually shown that many of these histone deacetylases are overexpressed in a number of cancers. And if you block using these small molecules, if you block the expression of some of these histone deacetylases, you can actually prevent cell proliferation, and some of these inhibitors of HDACs, these are called as HDAC inhibitors, they can actually serve as anti-cancer agents. And in fact, very surprisingly, some of these molecules have even been approved now for human use for certain treatment of certain kind of cancers. So this concept that histone covalent modification of histones can affect gene regulation has now led to the proliferation of a number of pharmaceutical companies which are now trying to develop either agonists or antagonists which can either affect the histone modification. People are trying to modify histone acylases, HATs. People are trying to develop inhibitors of HDACs, and depending upon what kind, whether HATs are active or HDACs are active in a specific kind of cancer, and by using such inhibitors, people can actually find, or now thinking you can actually use this basic research of or the knowledge arising out of the histone covalent modification for the benefit of mankind, and you can actually develop molecules which specifically inhibit some of the histone post-translational modification. And this entire concept that histone modifications can actually, which is the histone modifications are at a level which is different from that happening in the DNA sequence. This is different from a genetic code. And this entire area in fact has now led to a new area called epigenetic regulation of gene expression. In fact, in the next class we are going to discuss what exactly we mean by epigenetic regulation of gene expression. And I am going to tell you that epigenetic regulation of gene expression is not really dependent on the linear sequence of DNA, it is actually dependent more on the histone code. And there is one more important event that plays a very important role for epigenetic regulation of gene expression, that is DNA methylation. So what we are going to discuss in next class is that how histone modification and DNA methylation can actually play a very important role in epigenetic regulation of gene expression. Okay. And at the end of the next class we are going to realize that all these things actually act in concerted manner. We have histone modifications, DNA methylations, all these things in concert can actually determine whether a chromatin should become a euchromatin or it should become a heterochromatin, or whether genes have to be activated or genes have to be silenced. I think I will stop here.